Hello everyone, is the slide visible? Please put a yes or no in chat box. Hello, is the slide visible, students? Okay, am I audible clearly? Okay, so good evening everyone. This is Devdatta Panda. So today we are going to discuss regarding cell biology. So today we will discuss regarding cells various contents. I hope you all have gone through the content I mentioned you 4 days back. So I hope everyone has studied. Has everyone studied? Tell me sincerely. I want to know. You have gone through it? You have gone through the chapter? Right? Siddhanta said yes. Okay. Lakshmi. Then others. What about others? Okay. Debasis also gone through it. Okay. What about others? Okay. Lokesh also. Okay. So I hope everyone has gone through the topic. So I'll be getting to know. Don't worry if you have gone through the topic or not. I'll be testing it during the class itself and I'll be getting to know how far you are telling truth or lie. Okay. Okay. So let's begin with the topic of today. Today we'll be studying the cell biology. Okay. So all of you know, as you have gone through the topic, the basic thing, first of all, what you know, that cell is the fundamental unit, right? Cell is the fundamental unit, smallest units of cell, smallest unit of living organism, which is capable of doing self reproduction, all the life function, whatever is required, it is capable of doing it, right? सो सेल क्या होती है? सेल सबसे छोटी चीज होती है जो कि अपने आप से सब कुछ कर सकती है, राइट? सो नेक्स्ट टू दैट we need to study that various you might have gone through various historical points, right? So all the historical points no need to remember, but you have to read it several times so that you will have an idea. At least if you cannot remember all the years, at least you need to remember the scientist name and what his contribution was. And the famous scientist name, as all of you know, for the every famous scientific name, for that you need to study, means the whole content, right? Okay. uh, so here, to mention a few only, I have given few scientists. Rest of the scientists you need to answer. So the very first scientist is, whenever we study the cell, like first, who is the person who has observed the cell first? This is the person, that is Robert Hooke, who has observed the cell in a cork tree. But what he has observed, that cork tree, when he observed that particular piece of cork, that was dead. Okay, so what he observed it, he observed a honeycomb like structure. Okay, only the outer shells he can see, nothing else, no cytoplasm was there, nothing else. Okay, then Leeuwenhoek has seen some, he has taken the scrappy from his tooth and he has seen some bacteria, some other microbes also in it. Okay, and after that I have given, this is the very very important point, that is in 1938. And not to means you need to mention 1939 also, Sheldon and Swan, they have proposed a cell theory. This is very very important. In which year they have given independently this theory, we will be studying this in detail in the next slide. Then Colliker has given the thing, that is mitochondria in muscle cell, in 1957. And then Koch postulate came in 1882. Then Knoll and Ruska, this is important. Here Ruska name is mentioned, Knoll is not mentioned. Knoll and Ruska has given the first transmission electron microscope. Okay, electron microscope is the very high powerful microscope. We can see very finer details of the cell using the electron microscope. Okay, that came in 1931. Okay, that was transmission electron microscope. And in 1965, first commercial scanning electron microscope came. Okay, both are type of electron microscope only, but in both before in the machinery and some of the parts we will see in the later slides. So from this slide, the you will be studying the microscopes and its detail, like parts of microscope, differences between the working principles of microscope in microbiology section. So I would like all of you to go through that section today itself from the book, that is a Taro and Kapoor book, um, Microbiology by Taro and Kapoor. In that book it is very well mentioned, that is the textbook, referral textbook for your microbiology also. For all the chapters you have to means all the sections you have to go through that book only. So initially five first five chapters and last five chapters are very very important. So I think in first or second chapter you will be getting all the microscopic things, like how the microscope is working, which microscope works on which principle, you'll be getting in first or second chapter. You have to go through today or tomorrow itself. Okay. Okay. Then this is the brief about the history. Now that you have studied, you told me, no, you have studied the things. So I'll be asking you a few things. Just answer me in the chat box, or I'll be unmuting you. Just answer me. Okay. Now you can unmute yourself and answer whatever I'm asking. Okay. So tell me. Um, okay. Can you tell me, among Shildon and Swan, who was botanist and who was zoologist? That is a very important question. They used to ask several times. Can you tell me? Anyone? Yes. Shildon was botanist. Shildon was botanist. Swan was zoologist. Yes, that is correct. Shildon was botanist and Swan was zoologist. Okay. Then Robert Hooke has a very famous book. Do you know the book of his? He authored a book. What is the famous book Robert Hooke has authored? Micrographia. Yes, yes, it is Micrographia. That book is very famous work. Okay, and in that book, Robert Hooke has given the for cell. He has given the term cellulae. Okay, right? CELLULLULLE. Okay, cellulae. So in that book, he has given term cell. He has used the term cellulae, right? Then another scientist is there who has coined the term for cell as sacules and utricles. Do you know who is he? Cellulae means nothing. It is see, before, like we used to say, means before a particular term is coined for the particular thing, before that they used to give some words from their original language, whatever language they are using. For cell itself, he has used the term cellulae. By that time, cell term was not coined. The cell term was not in use. So he was the first person who has seen some kind of thing. Okay. For because of that, he has coined this, used this term, cellulae, right? So I asked you, like, who has used the thing like sacules and utricles for cell? Okay. Do you know the scientist name? No? Okay. Malpighi. Marcelo Malpighi, you know, right? One scientist, famous scientist, Malpighi and Tubal. You might be definitely knowing Malpighi and Tubal. Yes, that Malpighi. Malpighi has given the term that is sacules and utricles for cell. Okay? For cell, he has termed this particular thing. Okay. Okay. For now, this much is okay. Let's move to next thing. Do you know who is this person? Anybody can identify? He is Robert Hooke. Okay. This is the apparatus. This picture you see, this is the apparatus where you can see he has, first of all, he has observed the cell. Okay. Cork cell. He has put the cork cell and he saw some box like thing. Just a second, students. So I was telling you this particular scientist, he is Robert Hooke. Okay. So he saw the thin slices of cork, and this is the apparatus used by Robert Hooke. Okay. So next, coming to the characteristic of a cell, when we can call a particular thing as a cell. So it should be very high organized. It should be have a highly organized molecular and biochemical system, and it should produce some energy and use some energy in some way. Okay. It should be capable of movement. Cells should have a particular orientation also, and it should be capable of movement also. Orientation in the sense it will be placed at a particular position only, and movement in the sense that it is capable of moving from one place to other, means it will not run for 10 kilometers or somewhere else, but as per the growth pattern is moving. Like say my for the kids, their height is growing, so their stomach is means initially their stomach, their legs are small, only 1 feet height, so their stomach is at 1 feet high. After that, when he will grow big to 6 feet, his stomach will be at 4 feet height somewhere, no? So everything has to move, all the cells has to move somewhere. So means in the way, what I want to say is, cells move in a manner, in a particular way of growth, and that particular pattern of growth is restricted in case of animals, and that particular pattern is not restricted, not much restricted in case of your plants. Plants will grow throughout their life cycle, and restriction is there based on the hormonal pattern and all those things, but they tend to have some sort of movements. Okay. Then they can sense the environmental changes. So environmental changes can be very well, sense of environmental changes you can see in case of your prokaryotes, especially like amoeba. It moves far away from your any chemical thing whenever it encounters. That is called, you might have heard the term chemotaxis, right? So that is the thing, that is that it can sense the environmental change, so there it can sense to chemical. Like otherwise also phototaxis, chemotaxis, there are several movements according to the particular environmental changes, right? Sorry. So cell can duplicate itself. Then cells should have a particular self regulation also, means cell, if the cell is going to be cancerous, so cell tend to be, means after a certain cycle cell has to die. Okay. Cell will lose its viability, so cell has to die. So when the cell will go for that process of death, that is also regulated via some mechanism inside the cell. Okay. That is also we used to find that particular thing in case of your regulation of cell cycle, right? So all these characteristics should be there for a particular thing to be called as cell. Then next thing is cell theory. Cell theory, that cell theory, what it states, there are three important points of cell theory. First thing is, all organisms are made up of one or more cells, and the cell is the basic unit of organism. All cells came from the existing cells. Three scientists here you need to remember: Schilden, Swan, and Virchow. So as I have already stated, Schilden was Matthias Schilden. He was a German botanist. This particular, for the important scientists, for some of the important scientists, their country of origin is also important. So we need to remember that Schilden was German and Swan was British. Okay. German botanist was Schilden and Swan was British zoologist. Okay. Schilden has given his theory independently in 1938, and Swan has given the theory in 18. Sorry, did I mention? Sorry, it is 1838, and British zoologist, that is Swan, has given his theory in 1839. Okay. They have given the first 2 points. This third point, that is all cell came from the existing cell, that is commonly called as a term, a phrase is given for that, that is called hominis cellula e cellula. He has given this particular phrase in some language, means in their own language, but this particular phrase sometimes comes in exam. So it was what it was given by Virchow. Okay. Later on, after this particular two things came, Virchow has given this in 1855. So after that, the cell theory, this particular thing, after addition of the third point, it is called as cell principle or cell doctrine. Okay. So these 3 points you need to remember. But there are several drawbacks, means there are several points that deviate the cell theory. Like virus cannot be explained by cell theory, and there are organisms who have the multinucleated mass of cells, and there are organisms like acetabularia who has a very differentiated body, but that is within a particular cell itself. Okay. Hello. I cannot. uh, Okay. So I'll show you the picture of acetobularia just now only. Just wait for a second. So the particular scientist is, this is Sheldon, this is Swan, and this is Virco. Okay. Sheldon was a botanist, Swan was a zoologist, and Virco, he gave the third point, that is omnicellula, ecellula, right? Okay. So before going for microscopes, I wanted to show you the picture of. Okay, acetabulary will come somewhere. I'll show you that picture. So you can see what is the peculiar thing about acetabularia is it is a giant algae. Okay, but it is made up of a single cell itself. So that debates that how come a particular organism which is a unicellular, but it is having such a differentiated body. So that is not possible. So that debates the normal theory of your cell theory. Okay. Next, to study the cells, there are several, the instrument which is getting used, that is microscope. Why? Because the cells are not visible in the naked eye. So microscopes are used where the various lenses are used. So the lenses will magnify the thing and you can see the things in detail, right? So the first name which comes in the microscope thing, that is Zacharias Janssen. He has made the compound microscope, and from the credit of simple microscope goes to Galileo, and the microscope used by Leeuwenhoek is a kind of simple microscope itself. So you can see this particular structure, you can see over here, this is the structure which was used by Leeuwenhoek. He is Janssen, Zacharias Janssen. So Leeuwenhoek used this particular thing to see the organism that was present in his tooth, Scrappy. Okay. Okay. So this is the modern light microscope you can see in your labs and all. It is binocular. Previously, we used to have, you might have seen in the patho labs also in for testing the blood sample and all. There you might have seen the older one that is having one ocular piece, but this is binocular, means two, you can see from both of your eyes simultaneously. And that previously that uses the light, normal sunlight as source, but here the electricity, means there is an inbuilt lamp over here which provides the light for viewing the sample. You have to place the sample over here and you can see. Okay, these are the various magnification labels you can see. This particular thing is the electron microscope, and here electron beams are bombarded on the particular sample, and you can get an image of sample over this monitor. Okay, so you don't have to see over here, means you have to standardize the thing. Here is the eyepiece. Here you can see the samples over here. You have to put inside the, means sample inside, and electron beams are bombarded from on the particular tube present over here, and you can see the image over this particular monitor and save it. Okay. So coming to, this is the another photograph of electron microscope. You can see over here. So here magnetic field vacuum thing is used. Vacuum beams are used, and a beam of electron is passed over the sample, and it is very very powerful one, and it is 1 lakh up to 1 lakh magnification. Means whatever the object size is, it can be magnified 1 lakh times using this microscope. Okay. So I have already told you the two types of microscope, that is scanning electron microscope and transmission electron microscope. So the scanning electron microscope has a property, it will produce a 3D image, and only the surface of the specimen is can be observed using this scanning electron microscope. Okay. Then in the transmission electron microscope, this is the image of a transmission electron microscope. It will produce a 2D image. This is the important difference between these two, scanning electron microscope and transmission electron microscope, that the scanning electron microscope will produce a 3D image. This will produce a 2D image. Okay. And here, all the inside parts also you can see. Okay. Okay. This is a scanning electron microscope from very close view. I thought I should put some pictures because yeah, in UG project means UG program you might have got to see these pictures, might have not. So I thought it will be a good thing if you can see the pictures. Okay. Okay. So electron microscopy has some advantage of its own, that electrons have shorter wavelength than light. Definitely that because of that reason it provides a high resolution. Okay. And the maximum resolution, whenever we are viewing a thing very closely, we can get the finer detail, definitely. But the disadvantage is that the specimen should be in a dead condition. Means we cannot use the live specimen when we are using the electron microscope. Okay. And the process involved preparing the specimens is damaging, means destructive one. You cannot use the live one. So even if we are trying to see some cells or cultures, we cannot use their electron microscope. Okay. So okay, so moving next to the cell types, that is we have two types of cell, basically prokaryote and eukaryote. If you just divide the cells broadly, you can see two types of cell, prokaryote and eukaryote. We all belong to eukaryote thing. And all the whether it is prokaryote cell or eukaryote cell, they have some certain properties. Like the size of the cell is confounded by several factors, like nucleocytoplasmic ratio. Nucleocytoplasmic ratio means how much nucleus can be possibly there for a particular size of nucleus. आपका nucleus कितना बड़ा होगा और उस पर्टिकुलर न्यूक्लियस साइज के लिए आपका एक सेल के अंदर कितना साइटोप्लाज्म रह सकता है. ठीक है. That decides the maximum attainable size of the particular cell, or else the cell size will go on increasing. That does not happen. That happens only in case of your cancerous thing, means there is uncontrolled cell division, cell has so many abnormalities. So there for a normal cell there is a particular nucleus cytoplasmic ratio. Okay. For what amount of nucleus cytoplasm, what should be the size of the particular nucleus. And cell surface area of the surface area to volume ratio. Surface area means if you just move your hand around the cell, whatever you touch, that is the surface area of the cell. But if you just carry the cell in your hand, whatever space it has taken in your hand, that particular thing is called the volume, right? So surface area to volume ratio of a particular cell is also fixed. It will not, it cannot never go beyond that particular size. So we can see here the diagram of prokaryotic cell and eukaryotic cell. Definitely eukaryotic cell has much more advanced machinery, advanced in the sense that there is a very high division of labor. What do you mean by division of labor? Can anybody tell me what is division of labor? Yes. Yes, please. See, you need to interact. This will not work. You have to interact with me. No, nobody knows. At least say no, that you don't know. Are you hearing to me? Anybody there? I told you from the very first day that you should not be idle in the class, right?
idle in the class, right?
Yeah, thank you that you are hearing to
Yeah,
me.
thank you that you are hearing to
And
me.
please tell
And
me, do you know regarding the term division of labor? Anybody? Yeah, division of some
yeah, division of
Bhagyalakshmi has
some
texted.
Bhagyalakshmi has
That
texted.
particular organelle will do
That
particular
particular
function.
organelle will
Yeah,
do
somewhat
particular
similar to
function.
that
Yeah,
only.
somewhat similar
That
to that
we
only.
have so much
But
work to do
we have
inside a
so many
particular
work to
cell,
do
right?
inside a particular
So
cell, right?
if
So if
all the organelle will go on
all the
doing
organelle
the
will
same
go
thing.
on doing the same thing,
Just a second. If all the organelle will go on doing the same thing, then it will be chaos, no? If all the persons in your family will do the all the things, they will do cooking. Means 5% will do cooking, 5% will do cleaning. Then it will be chaos, no? So in a particular family, we have persons like mother will do cooking, we will do some assisting work, or we will study. Father will bring vegetables. Whatever, every person has some particular work, so all the work cannot be performed by a particular thing or particular object. The same thing over here. In prokaryotic to eukaryotic cell, there is a jump, and this particular jump is division of labor. That the labor means the work of whole cell is very meticulously divided to different organelles. Okay, whereas in case of your prokaryotic cell, you can see one organelle, one part. Let's say for example, this particular thing, that is a fimbriae or pseudopodia kind of thing, or the cell membrane, it has to do so many things. It has to protect the cell also, it has to do the excretion also, it has to do some sort of respiration activity also. Several things it has to be done by the particular membrane. But since it is only one cell structure, so it can manage to do the thing in particular this particular manner. But we are multicellular, no, we cannot do all the things by our membrane. So there are particular organelles in our body which do separate functions for our cell. Okay, coming to the next thing, that is, yeah, I was telling you, no, I'll show you the photograph of acetobularia. This is acetobularia. Just a second, students. This is acetobularia in somewhat enlarged view. So you can see over here this particular thing, means this particular leaf like thing and the stem like thing over here. This is comprising the one particular single cell only. Because it is zoomed, it may look like somewhat bigger, but it is also a bigger kind of thing, but and it is quite a differentiated one. Okay, acetobularia is algae. Okay, sea algae. So you can see the structure present over here. That time I was telling you, no, sorry that I have put the slide over here. So you can see the differentiated structure of acetobularia present over here. So coming to the thing which we are discussing, that is prokaryotes and eukaryotes. So in prokaryote, eukaryote, there are several differences, and whichever I have not mentioned over here, you need to tell me. I'll ask. Okay. So first one is DNA. So the DNA is not organized in a particular sort of nucleus in case of a prokaryote. So that is present like freely in the cell. That is called as nucleoid. That is a thread like thing, no nucleus sort of thing is present over here. Here there is a very prominent membrane bound structure present in eukaryotes, which encompasses your chromatin material. Okay, the size is usually smaller. Here the size is quite big. Here the organization is usually they are single cell. It is often multicellular. And here metabolism, if you will see, you can see over here the oxygen rarely needs to go inside. It will just touch the cell and go. Means by touching the cell itself, cell surface itself, the cell can take the required oxygen. But for us, oxygen needs to go inside the cell, then only we can do the respiration process. Okay, and there is no membrane bound organelle found in case of your prokaryotes, and quite a high number of membrane bound organelle you can see over here. So now you tell me, hmm, okay, now you need to tell me the other differences which you have studied between the prokaryote and eukaryote. I'll be asking the points whether they are present in prokaryote or eukaryote. You need to tell me. Okay. Quick, just make it quick because we don't have that much time, right? We have to finish so many things. So make it quick. Okay. So yes, ribosome. Ribosome is present in ribosome is present in both. Okay. Cell division occurs by budding or fission in case of your prokaryotes. Yes, histone is absent in prokaryotes, correct? Then what about nucleolus, guys? What about nucleolus? Yeah. Speak first. Absent in prokaryote, correct. Then in case of your eukaryotes, plasmids are seen. Plasmids are seen? No. Plasmids are not seen in case of your eukaryotes. It is seen in case of your eukaryote. Okay, I got it. Then what is plasmid? Anybody can tell me? Any idea what is plasmid? Circular DNA. Yes, it is a circular DNA, but it is extra chromosomal. Okay, it is, yes, extra, not, it is not extracellular, it is extra chromosomal. I'll tell you here I found a confusion. So it is not extracellular, extra, okay, extra nuclear and cellular, there is a difference between them. Extracellular means out of cell. Does the plasmid present outside the cell? Is it so? No. It is present inside the cell, but it is not part of the original genetic material. That is, it is not part of this particular nucleoid. It will be present over here, somewhere over here, and it is separate from the your genetic DNA, right? So it is extra chromosomal. The term is extra chromosomal DNA, right? So is it clear now? What is plasmid? Plasmid is a extra chromosomal DNA. Okay, we will get to see the plasmid in detail later on. Then next point. Cytoskeletons are present. Cytoskeleton, regarding cytoskeleton, what happens in pro and u? First, chromatophore is present in prokaryotes. Yes. In eukaryotes, cytoskeleton is present. In prokaryotes, cytoskeleton is not there. Okay. Then regarding ribosomes, which kind of ribosome is seen in prokaryotes and which kind of ribosome is seen in eukaryotes? 70S is present in pro, yes. 80S is present in eukaryote. Okay. Then regarding transcription translation, do they occur simultaneously? Simultaneously, it's occur in case of your pro or u. Transcription and translation simultaneously occurs in case of your prokaryote. We'll be seeing this point in case of your transcription translation thing, that because all the things happen inside the cytoplasm itself. So in case of your prokaryote, after the as soon as the RNA is getting synthesized, it is involved in translation process. But in case of your eukaryote, there is some processing required in case of your RNA before going on for protein synthesis, right? Okay. Rest of the points you need to study. So I have given here some points. Most of the points you already have told. Is there any point remaining? No, most of the points you have told. Congratulations. I thought I got to know that you have studied some points. Good. Yeah, centrioles. Yeah, tell me regarding centrioles. Yes, Siddhant. What about centrioles? Do you know in prokaryote centrioles are present or not? Centrioles are present in eukaryote, that too in case of your animal, right? In case of your plants also, centrioles are not present. Yeah. Based on flagella, cilia flagella are basically present in case of your prokaryotes. Flagella are not basically part of eukaryote, but in some eukaryote you can see some sort of flagella kind of thing. Okay, in some of eukaryote only, but that is basically a feature of your prokaryotes, right? Okay, let's move to the next thing, or else it will be very detailed one if I keep on discussing. So rest of the points you study, wherever you find any confusion, we can discuss, right? So here you can see also flagella. Here also I have mentioned flagella. It is the property of both, but basically it is found in case of your prokaryotes. In some of the eukaryotes also you can find this thing. Okay. Okay, coming to the next thing, that is what is the difference between the plant and animal cell. I have enlisted some of the points. That is, plants have the plastids, animals do not have the plastid. Regarding cell wall, here it is made up of cellulose. Animals do not have a cell wall. And here the vacuole is present. Vacuole is the characteristic, one of the characteristic feature of the plant cells. Whereas in animal cell, mostly vacuoles are absent, or if present in case of some organism, it is very small. Okay. Sorry students, I forgot to ask you, are you getting the speed I am talking, or you want somewhat slower, or okay, no? Is anybody is having problem with understanding my language? Yes, please pick up if you have any problem. Okay, thank you. So next is, plants may have plasmodesmata, but animals do not have plasmodesmata, and plants do not have centrioles. Here the all the spindle thing is formed from asters. Astral ray we'll study later on. From the both the poles, it will form the centriole. It will form the spindle fiber. Here the centrioles are present. Here the cell do not have cholesterols in the membrane. Plant have some other things. Ergosterol is a component of plant membrane. Cholesterol is not there. So mostly you used to say that uh, and the cholesterol, we'll see that in the membrane section. And next thing is that animals have cholesterol in their cell membrane. Okay. And the excess glucose is stored in the form of starch. Here the excess glucose is stored in the form of glycogen. And here the shape is completely fixed. Why? Because the cell wall is there, which is rigid in nature. But this is mostly amorphous cell, because cell membrane is there, no cell wall is present. So it will, it can means have different shape, right? So that is the difference between plant cell and animal cell. And okay, you need to tell me the rest of the difference. What are the difference I didn't mention, please? Yes, tell me. What about cytokinesis in case of your animal and plant cell? Yes, answer quickly. What about your cytokinesis? Yes, furrow method occurs in case of your animals, and in case of plant cell you can see the cell plate method. Okay, then yeah, cell plate method. Then glyoxysomes are completely absent in case of your, in which case glyoxysomes are absent, plant or animal? Answer fast. Animal. Glyoxysomes are absent in animal, and in which part of where you can see in plant, where you can see the glyoxysomes role most? Do you have any idea? Glyoxysomes are basically present in seed. Okay, in plant seeds glyoxysome has a important role. Yes, yes, the bashes you have mentioned, it is seed only, correct answer. And what is the specific term? Golgi body is seen in both the cases, animal and plant. But in case of your plant, there is a specific term used for Golgi body. Do you know what is that? Yes, dictyosome. So what is difference between dictyosome and Golgi body basically? So in case of your plant, the Golgi body is interconnected. One Golgi body is interconnected to another. So they form a stack kind of things. In that condition, it is called as dictyosome. In case of your plant cell. Yes, correct. And hmm, chloroplast. Chloroplast is present in, no need to say, it is chloroplast is present in case of your plants and it is absent in case of your animals. Yeah, correct. Good. So that is all about a plant and animal cell. Is there any point you want to mention apart from this? Yeah, please. Okay. No. Okay. So let's move to the next thing, that is chemical nature of the cell. What is your cell is composed of? So cell is 70% of the cell. 70%, you might have heard, the 70% of human body is water. So human body is also made up of cells, no? So water comprises a maximum portion of the cell, and whatever thing you can see that are suspended in the matrix that made out of water. So apart from water, you can see various chemicals like carbohydrates, lipids, proteins, and nucleic acid, and some minerals, some ash inside the cell. So carbohydrate, you know the function. Carbohydrate is a source of energy, and carbohydrate, that is a glucose, which is used in respiration process to produce the ATP, right? In what, where the ATP production occurs, using the, means your glucose as a source, which organelle is involved? Yes, please answer fast, fast, fast. Mitochondria, yes. So next is lipids. Lipids serves as the energy storage, and lipids, what it another function is, it reduces the heat loss. Okay, in the winter, fatty persons who are more thick, जो मोटे होते हैं ना, उनको कम ठंड लगती है क्योंकि उनके स्किन के अंदर बहुत ज्यादा फैट रहता है ना. तो बुरा मत मानना कोई भी कैसा. आई डोंट मीन टू कमेंट ऑन द शेप, don't get me wrong. I just wanted to lighten up the mood. Okay, so lipids are act as an insulator under the skin and reduces the heat loss. And next thing is proteins. Proteins are build up, they used as the building blocks for the growth and repair of the organism. And they are, proteins are, means all the enzymes mostly composed of proteins only, apart from some of the what exceptions like ribosome and all. Okay, then nucleic acid, as you all know, it is a DNA and RNA which carry the genetic information. This slide is important from your JRF point of view also, and other examination point of view also. So the sodium ion is important. Why? Because it takes part in the transmission of nerve impulse, and that is in case of your animals. In case of your plants also, it maintains the osmotic balance and participates in so many factors. Sodium and calcium, potassium, these three elements are very, very essential for signaling mechanism. Signaling mechanism means whenever we are sensing something or plant is sensing something, sensing means it is responding to some external signal. It depends on several messengers, right? So they act as a part of the signal transmission system. So they are very, very important. And potassium also part of the nerve impulse transmission, and they also maintain the osmotic balance. Calcium takes part, it is a chemical component of teeth and bone. No need to remember. Basically calcium ion is important for several reactions occurring in your mitochondria and chloroplast also. Okay, and apart from that, iron, you can see iron is a very important part of your etc, that is electron transport system. There iron plays a very important role. Magnesium is present in case of your magnesium, where in which particular thing which is very important to plant cell, magnesium ion is present. Can anybody tell me that molecule is very important for plant? Yes, it is chlorophyll. So in chlorophyll, in the center of the chlorophyll, that is the porphyrin ring head, it is present. Yeah, calcium is part of your middle lamella. Calcium and magnesium both are present in case of your middle lamella. Yes, it is correct. Then the next is Nitric oxide, nitrate ions, nitrate ions are important from your biology nitrogen fixation point of view, that in which form your plant is taking the nitrogen, and ultimately what the nitrogen will be used for. Nitrogen are the part of your nitrogenous bases, right? Nitrogenous bases, why we call it as nitrogenous base means here the nitrogen, they are basic in nature and nitrogen is present over there. You might have seen the purine pyrimidine structure. So there in that is the nitrogen is present in the structure itself. So nitrogen has the important function in case of your inside cell, that is it is a part of your nitrogenous base. Phosphate ions are very very important. Why? Because they are the part of ATP. ATP is otherwise called as, yes, answer fast. ATP is otherwise called as, yeah, tell me faster. Nobody knows. Yeah, it is. Pile has answered. ATP is the energy currency of the cell. Yes, it is adenosine triphosphate. Where the energy is stored in ATP? Anybody know? Means how ATP is giving you energy. ATP means adenosine triphosphate, right? Some molecule in the structure, if you will see the ATP, some molecule means major molecular structure is over here. Side to it, 3 phosphate groups are attached. One phosphate, second phosphate, then third phosphate. So with breaking of one phosphate from this particular molecule, it will release energy. ATP, when it releases first phosphate, it will become ATP. ATP will become ADP, adenosine diphosphate. Then it will release another phosphate, it will become adenosine monophosphate. Okay. So in the energy currency, if you'll see, ATP is the highest energy carrying molecule, then ADP, then AMP. Okay. So that's why this phosphate thing is very important. And another thing is that phosphate is important in your signal transduction process, where molecules are activated or deactivated using the particular molecule, that is ATP or other energy currencies. Okay. Okay. So this is about the chemical nature of the cell. Moving to cell organelles. Okay. So you need to remember, means if you just mug up the thing, you will never remember. Okay. Means even if you remember now also, you will forget later on. So you need to study it in a very light manner. Right. Yes. Phosphorylation. Yeah. Did I use the term phosphorylation? Phosphorylation is a process that is where a cell, where ATP molecule is getting added. That is the process called phosphorylation. Okay. It is here the enzyme used is kinase. Kinase enzyme, what it does is whatever molecule is present, it will go and add a phosphate molecule to it. So phosphorylation, whatever rylation and the prefix is used, that thing is getting added, right? So in that way, carboxylation means carboxyl group is getting added to your molecule. This is a molecule. Okay. To that your phosphate group, phosphate means PO4, PO4 minus 3, this group is getting added to the particular molecule. Okay. So it is getting, this molecule is getting energized, and whoever has donated this particular thing is losing its energy, right? Okay. So okay. Okay. So let's move on to the organelles. Yeah. So here the important points I have mentioned. First one is cell membrane. Cell membrane acts as the security gate. Means it will check what things will enter, what not. Okay. It is like a big colony, or you can say big apartment kind of thing. There will be something who is controlling the whole apartment. Then only the apartment is functioning, or people are staying in peace, right? People here is the cell itself, the big fat people who is getting all the peace and getting the service from all these things, is the cell. And ultimately we are getting enjoying the livelihood, right? So here what organ does what? First thing is security gate. This cell membrane act as a security gate. Second thing is control center. Control center means it acts as the brain of the cell. It controls everything, that is the nucleus, Next thing is assembly line, that nucleus will instruct the particular thing. Nucleus via your chromatin material, it will tell the cell that okay, now I am producing this particular thing. So whatever thing is produced, it will move in this particular passage. You might have seen in the various games, like one rabbit's home is present over here, and rabbit is sitting over here. It has to move some passage to reach the home. So you just imagine the endoplasmic reticulum, that kind of structure. Okay, so it is a pathway, a passage through which the cell, whatever material produced in nucleus, it has to move, right? So how can it move? The nucleus had some pores. These are the pores from which the RNA will be produced by the cell, and it will be coming to the outside. So the endoplasmic reticulum is also called as assembly line. Then after the things are coming from RNA came to the cytoplasm now. So what it will do? RNA along with the ribosomes present over here, that is the workbenches, they are sitting in the cytoplasm, they are sitting on the rough endoplasmic reticulum. What they will do, out of that particular RNA, it has to be processed further to produce the what? Protein. Then they will collaboratively produce the protein, and other things. Here in the endoplasmic reticulum, proteins will be produced and fat will be also produced. Whatever things are produced in the cell, next thing they have to move through this particular structure. What is this? This is Golgi complex, right? This is Golgi complex, and this act as a distribution center. Means it will further process the molecules, and later on, whatever material is required by the cell, it will be there in the cell, and it will be finally used. And whatever thing that is a torn out thing, it will form the lysosome thing, and the lysosomes will ultimately move out of the cell. Here you can see the cleaning crew, that is the lysosome. That is what they do. Whatever material that is unnecessary for the cell, they will engulf the particular material, and ultimately they will digest it, and the whatever thing digested material, either they will be reused by the cell or they'll be excreted out, right? Then for all these things we need energy or power. So for that, mitochondria is doing the task, that is it is producing the power. And to hold all these things together, this particular thing, that is cytoskeleton, is present. Cytoskeleton is like what you can say, there is a mess, there is a, in sofa you have seen the mess, means wired mess. सोफा में आपने देखा होगा, ऐसे ऐसे वायर्स पड़े होते हैं, ऐसे क्रॉस क्रॉस, इधर से उधर, उधर से इधर. So what the they are thin itself. If you sit on one wire, you will fall down and eventually wire will break. But if they are collectively together, 100 of the wires and 100 of the threads, they have some means they get some um, tensile strength. So based on that strength, they support the cell. Okay, so this is in brief. Yeah, they also provide the rigidity of the cell. Basically comes from your cell wall. In case of your plant cell, yes, in some sort you can tell that it is providing rigidity, because if the cytoskeletons are not present in the cell will become you just imagine it like um okay, you just imagine it like a tent. You have put a tent while going for camping and all, you used to put tent, no? In the tent you are putting some rods, and above all you are putting some cloth, and that to make a temporary home, right? So if you just remove the rods, what will become? That home will collapse, no? That tent will collapse. So in that way it provides a rigidity. Just compare the thing. Trafficking and motility is related to cytoskeleton. Trafficking is basically related to, yeah, some sort of trafficking is done by cytoskeleton. Means it helps to movement of means on the cytoskeleton, some things are moved from one place to other, but basically the molecule trafficking is done by your endoplasmic reticulum and Golgi body. Okay, motility, cytoskeleton is responsible. Yes, it will extend itself. We will study cytoskeleton in detail, don't worry. I was just giving you a brief idea regarding all the things. So you just imagine cell like a factory, so you will get to know. Okay, before going for cell wall, tell me something about protoplasm. Yeah, have you studied protoplasm? Okay, so tell me, who has termed the protoplasm? Who coined the term protoplasm? Purkinje. Yes, it is correct. Purkinje has coined the term protoplasm. It's correct. Then protoplasmic theory. One theory is there called protoplasmic theory. It was proposed by, do you know who proposed it? Yes, protoplasm is cytoplasm plus nucleus. Yes. So that particular theory, that is protoplasmic theory, it was proposed by Skulge, Max Skulge, Schultze. I hope you are having a pen and paper and writing whatever I'm saying. I hope you are having that, right? So write it down, the scientist name, Skulge, Schultze, Max Skulge. Okay. So he has given the protoplasmic theory where he has stated that all the living cells are made up of protoplasm in which all the vital activities occur, right? Okay. Then that is about protoplasm. So protoplasm is consisting of your cytoplasm and nucleus. Okay. So out of total cell, if you just remove, if you just let me show this picture. Okay. In this particular cell wall is not there. Just imagine a plant cell. So if you just remove the plant cell wall, whatever remaining, that thing is called as protoplast. Okay. Your cytoplasm plus nucleus, that is called protoplast. And if you just remove the this membrane, that cell membrane, apart from that cell membrane and nucleus, if you just remove whatever remaining inside organelle plus the liquid matrix, that is called as your cytoplasm. Okay. Then in cytoplasm, the liquid portion is called as, spelling again, okay, SC. Okay, wait, I'll try to write it down. SCHULTZE. Scolge. Okay. SCHULTZE. He has coined the term, that is protoplasmic theory. Yeah, hyaloplasm, I'm coming. Wait. So hyaloplasm is the liquid portion. If you remove the organelles, after that organelles and whatever metabolically inactive substance, if you just remove all those things, whatever liquid part is remaining, that is hyaloplasm. Okay, plain liquid portion. So it is like from your sharbat, if you just remove the sugar, salt and whatever fruits you are adding, the plain thing, whatever remain, that is your haloplasm. It will be definitely transparent, no, because whatever color it is getting, that is because of the metabolic substance what present over the cell, right? So that is haloplasm. And what else is remaining? That is the cytoplasmic organelles. So protoplasm minus organelle. Hmm. I'll tell you, okay? I'm not getting the exact term. I'll tell you in your WhatsApp group. I'll tell you. Don't worry. Okay? Don't worry. I'll tell you. Okay. So this is about the thing. Hyaloplasm can be written as such. Protoplasm minus organelle. Not exactly. Hyaloplasm can be written as cytoplasm minus organelle minus metabolically inactive substance, that is your fats, oil bodies, all those things. Okay. You cannot write the protoplasm hyaloplasm as protoplast minus, because nucleus also need to be removed. No, if you want to write it in terms of protoplasm, you have to write it like protoplasm minus your membrane, cell membrane, minus nucleus, minus organelles. Then it will become hyaloplasm, right? In that way, it will become hyaloplasm. Okay. So let's move to next thing, that is cell wall. Cell wall is very important. So, so regarding cell wall, some of the important points I have mentioned over here. In plant cell only you can see this. First, from inside to outside, you can see the outer one. The outer one is, yes, which one is outer among this? Tell me. Among secondary wall, primary cell wall or middle lamella, which is outer one? Yes, middle lamella is the thing that connects between two cells, right? Inside, from outside to inside, you can see, first of all, middle lamella will be laid. After that, towards this, first this green one, that is, sorry, the blue one, that is middle lamella. Inside of that, you can see the primary cell wall. Inside of that, at last, this cell thing is formed, that is secondary cell wall. Okay, secondary cell wall might also having several layers, which is shown over here. That is basically seen in case of your woody plants, that several secondary cell wall layers are also formed. So basically your secondary cell walls are composed of cellulose, lignin and suberin. Because of that it has great strength, and it forms the wood, the wood out of plant which we get, no, that is the secondary cell wall mostly. Primary cell wall is composed of basically cellulose and some sort of hemicellulose is also present. And in middle lamella, basically pectin, magnesium and calcium ions are present. Okay. Okay. Then another thing which I want to mention regarding primary and secondary cell wall, you might have heard this term, intussusception and aspiration. Do you know this terms? Okay, I'll be telling you. So these two terms are related to the growth of the primary cell wall and secondary cell wall. So intussusception means here what will occur is the whatever material is growing, no, whatever material is getting deposited, um, it grows on the existing one. Okay, it grows on the existing one means the first thing is primary cell wall is thin and amorphous in nature, and incorporation of more cellular, first one thin layer will be formed. After that the other materials will get added to the particular layer, and in that way the primary cell wall grows. That is the process called, growth process is called intussusception. Okay. But in case of your secondary cell wall, what occurs is here process is aspiration. So in aspiration process, it is not like from inside the material will not come. They will form in a stepwise manner. So you just go through these two terms, and if you find the confusion, because I'm going short in time, so I'm not explaining this term in detail. You just go through these terms and read about it. If you find confusion, I'll be telling you in detail later on. Okay, and so I'm moving because we are in some in beginning only. So because in your organelles there is much of the things you already know. Okay, so this cell wall is totally non-living. Always and always remember, it is non-living, and it is produced and maintained by the living protoplasm, which is putting the things over the membrane. Okay. Then the functions you all know, it provides a cell shape, size, mechanical support, transport of substances, etc., etc. Okay. Another fact I want to share with you, in case of your gymnosperms, you can see there is a structure which is having xylans and mannans deposited. Xylan and mannan, two types of chemical, like suberin and lignin, no, it is also a metabolite. So there, in case of your gymnosperm, sometimes you can see tertiary cell wall also. That is an exception kind of thing. Okay. Okay. So moving to the next thing, that is your plasma membrane. Okay. So Tom cell membrane was given by Fast. I have already written over here. Blower has given this term, and it is semi-permeable in nature. Why? Because it is allowing some of the chemicals to enter, and not it is not allowing others to move freely. Why? Because of its chemical nature. It is composed of, the plasma membrane is composed of bilayer, lipid bilayer, and on this bilayer of lipid your proteins are embedded. Okay. So the lipid bilayer looks like something like this. It will have it will have a polar head, means hydrophilic head. Okay, hydrophobic tails present over here. Hydrophilic means, philic means it loves water. So this surface can face water and this surface can face water. These chains cannot, means they are hydrophobic, so they cannot face the water. So they are arranged like this. This is one lipid layer and this is one lipid layer. So the plasma membrane is consisting of two lipid bilayers, means lipid layer, two lipid layers or lipid bilayer it is consisting of, where hydrophilic heads are towards outside, hydrophobic tails are towards inside. So because this particular middle section is hydrophobic in nature and it is lipid in nature, so lipids can easily pass through your cell membrane. Okay, but the and whatever material has to come this side to this side or this side to this side, it has to go through this particular portion, no? So so the lipids can easily pass. Okay, but when it comes to the polar substances which are hydrophilic in nature, they can come and attach to this, but they cannot pass through this particular thing. Why? Because hydrophilic things cannot pass. Polar dissolves to polar, means like dissolves to like, as we have studied. In polar substances will mix easily with the polar things and non-polar will mix easily with the non-polar thing. Okay, so the hydrophilic substances like water or several carbohydrates, they cannot move directly through this particular cytoplasm, a particular membrane. Okay, they need some sort of transporter molecules to pass through this particular thing. Okay, and there are several models have been prescribed for the cell membrane. You know regarding them, just tell me the names of the models and who has proposed the models. Hello, hello. Tell me the names of the plasma membrane models. Daniel Dufson has proposed which one? Lipid protein. Daniel Dufson has proposed the trilamellar model or sandwich model, where he has mentioned that this is the first trilamellar model, and they have mentioned that bilayer of lipid lies between two layers of protein. So outside proteins are present and inside the lipids are present. Like that, he has stated the model. Okay, and fluid mosaic model याद नहीं आ रहा. ओके, कोई बात नहीं. So next model is unit membrane model that was proposed by Robertson. Robertson has stated a much more elaborate than your trilamellar model. So in Robertson's model, he has stated that um, in your membrane is a asymmetrical thing, and on the outer surface some proteins are there, and inner surface some other proteins are there, and the middle section is composed of your phospholipid bilayer. So it is somewhat similar to that of your trilamellar model, but in trilamellar model he has stated that there are three different layers, but in case of your unit membrane he has stated that three layers are present, but they act together, means they are in close contact to each other. Okay, means the structure and the model he has given, that is bit different in both the things, but the dimension given in the both trilamellar and unit membrane model is same only. They both have stated that membrane 75 angstrom in size, and the most accepted model is your fluid mosaic model that was proposed by, who has proposed the model fluid mosaic? Singer Nicholson, yes. Singer and Nicholson has proposed this model in 1972. It states that the membrane is semi-fluid in nature, and fluid mosaic. Why it is called so? Phospholipids. Uh, Phospholipids are movable thing, so it acts as a semi-fluid. So in that way, it is a fluidic nature. So for fluidic nature of the membrane is because of your phospholipids present on the cell membrane, and mosaic term is given because of the proteins are intermittently present over the cell. Proteins are present over here. So proteins are intermittently present, and because of that they are telling that protein icebergs and protein ice cubes are floating in the sea of your phospholipids. In that way it is fluid mosaic model. Okay, okay. Yeah, So here I have mentioned some points regarding the fluid mosaic model. So I cannot go through all the points because I'm lacking in time. You can see this thing later on also in video. Okay, that thing I already explained. No, that thing only they are telling. So the proteins which are present over here, totally outside, they are called. Here the extrinsic, intrinsic is not there. Okay, I'll be sending you all these questions. I have planned to ask in class, but time is not there. So please excuse me. I'll be giving you all this question later on for your study. So there are proteins called intrinsic protein. Intrinsic protein means that is completely embedded inside the membrane, and the protein which is completely outside like this, they're called extrinsic protein. And some proteins which span through the membrane. Okay, so these are some points regarding the cell membrane, extra points. So the composition is mainly because of the protein and phospholipids present, and some proteins act as a receptor and transporter, and lipids can move laterally in the membrane. Lipids, basically the phospholipids, they can move laterally, that is at a rate of 2 micrometer per second. And saturation of the lipids vary in the particular temperature and environmental condition. When the temperature drops, the organism will have more unsaturated fatty acid in the membrane, and as the temperature increases, the fluidity of the membrane increases. Why? Because lipids will become more liquid in high temperature, right? Then coming to the various cellular transport associated with your membrane. So there are two processes like active transport and passive transport. So active transport does not use any. Active transport uses, sorry, this is by mistake written. Active transport is uses energy, but passive transport does not use any energy. So for example of passive transport is diffusion, facilitated diffusion and osmosis. And the active transport part is protein pumps, exocytosis, endocytosis. All these processes are part of your active transport. Okay, so in active transport, the major thing is from low concentration to high concentration, the things has to move, so it will need energy. But in case of passive transport, from high concentration to low concentration it has to move, so it will not require any sort of energy. So it will be easy. Passive transport is easy, natural process. But active transport, it will require some energy to do this uphill journey, right? So in diffusion, molecules move from high to low concentration, so it's natural. Then osmosis, movement of water. In diffusion, all the molecules will move. The solute molecules will move. In osmosis, water moves actually. Okay, that is the basic difference between osmosis and diffusion. So here the water will move across the particular membrane. So water used to move from low concentration to low, means wherever the solute concentration is high, towards that direction your water is moving. Okay, that is osmosis. And then facilitative diffusion, it is the process where the diffusion is assisted by some of the protein molecules. Okay, so some carrier molecules are involved in this, so that is the process. Then this table is a summary of your various transport mechanism. This is very important. All the things I mentioned, that is present over here. So in case of your see, the simple diffusion is for lipids. Lipids can easily pass because lipids means the membrane center is hydrophobic, no, so lipids can easily pass. But for passage of proteins and other things, you need facilitated diffusion or active transports. And here in facilitated transport and active transport, you need the transporter to be attached to the molecule, then only it can pass. In diffusion, no need of any carrier molecule. And regarding energy source, I already told you. And the direction of transport also I told you. From this will be from high to low concentration, this two will be in the opposite direction. Okay, sorry, facilitated diffusion and simple diffusion both will be from high to low concentration. Active transport will be from opposite direction, though to high concentration it will move. Okay, then these three molecules are important, that is glycoprotein. Glycoprotein helps in recombination. recognition of the cell. Glycolipids present over the cell membrane, they also help in recognition of the cell. The next thing is cholesterol is a molecule which maintains the fluidity of the membrane in case of your animals. Similar kind of lipid molecules are present in case of your plant cell also, which maintain the fluidity of the membrane. Okay, these are for your some extra information. You can go through them later on also. So the membrane proteins, what function they used to do. Coming to the next term, that is nucleus. Okay, coming to the next term, that is nucleus. So nucleus was first observed by Robert Brown, and just give me a second. Yeah, nucleus was first discovered by Robert Brown in root cell of orchid, that you know already, right? And inside your nucleus, you can see it is the double membrane structure, and nucleus is having all these structures. That is, it is having a nuclear envelope or nuclear membrane, and nuclear plasm is also there, and nucleolus is there. Nucleolus, and inside nucleus this organ is present, that is called as nucleolus. It is not actually an organ, it is an inclusion that is found in the secondary constitution of your chromosome. Now besides to that, which is an area itself, but to specify, because it has some specific function, so we denote it as a new organelle itself, but it is not having particular shape or something. Then chromatin materials are dispersed throughout the nucleus. Okay. Okay. So I don't want to rush the thing. We can discuss the rest of the organelles in the next class, no problem. Because yes, we have studied the cell from several, starting from 6th or 7th we have studied. But the thing is that till now also, if somebody is asking you questions in the cell biology, only few persons are able to answer it correctly, right? So I don't want to rush, because it is already going to be 7 o'clock. So I am stopping the today's session here, but we'll be having some points. So you need to right now get ready to interact with me. The last 10 minutes I'll be asking you questions. Okay. Okay. So type in chat box or unmute yourself and answer quickly. We'll be having a fast, we'll be having some quick discussion regarding questions. Okay. Okay, you can unmute yourself and answer, or you can put the answer in chat box also. Okay. So the first question goes like, okay, tell me the subunits that are present in 70S ribosome.
50S plus 30S.
50-30. Then what about 80S?
60-40.
Okay. Next question is, I already told you who coined this phrase, unicellular, acellular.
Rudolf Virchow.
Others, please answer. Yes. See, nobody is going to eat you up. If you unmute your mic and tell, nobody is going to eat you up. So please unmute and answer. Okay. What do you know about the endomembrane system? What is that? Yes, anyone?
Ma'am, vacuoles, Golgi apparatus, endoplasmic reticulum, and lysosomes.
What is that? Means why they are called as endomembrane system? Yes. So to endomembrane systems are related organelles, means their function is related and they work in a coordinated way. So you see in the structure of, I have not yet discussed, but because you have read something, so just to brush up your knowledge and whatever you studied, you don't forget. I'm discussing 5-10 minutes questions. So I hope you can answer. So endomembrane system is the system. There are some of membranes. Okay, they are interlinked and you see in the structure of ER, then Golgi body, lysosome, vacuole, what it is actually consisting of. They are some of the membranes. That's it. Inside of them, something is moving here and there. So their function is interrelated. Whatever molecule is coming to ER, it will eventually go to Golgi body, then it will go to lysosome, or either it will be stored in vacuole or it will be digested by lysosome and getting out of your cell, right? Okay. The next question is, uh, who has given the term nucleolus?
Robert Brown.
Nucleolus.
Nucleolus.
Just a second. Nucleolus term was given by Baumann. Okay. And it was discovered by Fontana. Nucleolus. Robert Brown has discovered the nucleus, not nucleolus. Okay. The next thing is, does RBCs have nucleus? Yes.
No. No, ma'am.
Yes. Any other example of the cells which don't have nucleus in plants? In sheep elements and tracheal cells, yes, they don't have the nucleus. Yes, correct. Then uh, okay, glycocalyx. Do you know anything about glycocalyx?
Yes, ma'am.
Yeah, please.
मैम वो बैक्टीरिया का होता है, वो सेल मेम्ब्रेन होता है आई थिंक. सेल, हाँ, बैक्टीरिया का आउटर लेयर होता है ग्लाइकोकालिक्स.
बैक्टीरिया, इट इज प्रेजेंट इन द बैक्टीरिया. ये बैक्टीरिया का आउटर वॉल होता है जो कि means if it is forming a loose sheath kind of thing, that is called slime layer. If it is tough or thick, it is forming a capsule. Okay, so glycocalyx, that will be helping in what recognition of the particular thing, means it is like sensory organ of the bacteria. Okay. Okay, then okay, the next term is, do you know about mesosomes? Mesosomes, where it is present, what function it does? Yes.
मेसोजोम बैक्टीरिया वगैरह में होता है, वो रेस्पिरेशन आई थिंक.
Yeah, mesosome is a infolding that is present in the bacteria. That is the normal cell membrane will infold towards inside, and because in bacteria mitochondria is not present, so it has to do the respiration using that mesosome. Okay. Then okay. Mm. Okay. Now tell me the organelles which are single membrane bound. Do you know? Organelles which are double membrane bound and single membrane bound. You need to list out them separately and remember. Okay. Tell me the single membrane bound organelles, please. Type in the chat box. Ribosome is totally non-membrane bound. There is no membrane around ribosome.
Ma'am, except mitochondria, chloroplast, and or
Yes.
except mitochondria and chloroplast, I think, and ribosome are non-membrane bound. And then the rest of are single membrane bound.
See, I'm telling you, mitochondria, chloroplast, that is plastid.
Huh.
Nucleus is also double membrane bound.
Double membrane.
Golgi body, endoplasmic reticulum. These are double membrane bound structure. Apart from that, lysosome, vacuole, cilia, flagella, microbodies. Microbodies include a spirosome and what else? Peroxisome. They are all single membrane bound. Okay. And ribosome and centriole, these two are non-membrane bound. Remember them. Ribosome and centriole, they don't have a membrane. Okay. Okay. Then uh, protein sorting is done by which organelle does the protein sorting or packaging of proteins? Yes.
Golgi body.
Any idea?
Golgi body.
Golgi apparatus. Yes, yes, yes. Good. Which organelle is called suicidal bag?
Lysosome.
Which enzyme is present in lysosome?
Mm.
Which enzyme is abundantly found in lysosome? Catalase. Yes, it is acidic kind of material is present. Phosphatase, yeah, some sort of phosphatase might be present. But basically, acid hydrolases are present. Acid hydrolases, acid phosphatase, acid hydrolases are present. Okay. So next is mm, okay. Who has just a second. Okay. Among the plastid, for the flower coloring of the petal, which plastid is responsible? Which plastid is responsible for flower color?
Chromoplast.
Petal color, which plastid is present? Chromoplast. Yes, it is chromoplast. Okay. Which among inside the cell, which organelles are called semi-autonomous in nature?
Mitochondria.
Chloroplast, mitochondria.
Mitochondria.
Yes, yes, please. Then. Okay. Do you know anything about endosymbiosis? Endosymbiotic theory?
No, ma'am.
Endosymbiotic theory is the theory that tells about the origin of mitochondria and chloroplast inside the cell. They are believed to have been inherited from means they are actually primitive bacteria which were sometime in the ancient time it was engulfed by an another big bacteria means another bacteria. So they become part of it and it has given some sort of advantage to the cell, so the cell inherited those things. So it is said that one bacteria has engulfed the chlorophyll having means green bacteria means one bacteria has engulfed the blue green algae kind of thing. So that prokaryote cyanobacteria is that it has engulfed which has the chlorophyll in it. So that further it has modified to form the plastids, that is the chloroplast. And there is the another bacteria which is not chlorophyll having but it has the power to reduce the oxygen. So that reducing bacteria got engulfed by another bacteria which ultimately become the mitochondria by the cell. Okay. Then someone has said me that catalase is present in case of your lysosome, but catalase is the typical enzyme which is present over peroxisomes. Why? Because catalase is an enzyme. Peroxisome, what is the function? Main function of peroxisome is it will degrade the hydrogen peroxide which is formed in the cell, right? So catalase does that function of degrading the hydrogen peroxide produced in the cell. Okay. Okay. So that's it for today's class. If you have any doubts anywhere, we can discuss. You can put in the chat box also. We can put in WhatsApp group also. From out to in perinuclear cell, perinuclear space is first, then cell membrane or opposite one. I didn't get your question, Bhagilakshmi. Please unmute yourself. Yeah, you are already unmuted. Please tell me, speak the question, what you are trying to say. I'm not
Yes,
getting.
ma'am. Yes, ma'am. I mean to say, while coming from outside to inside, the first, the membrane means
Yeah.
the list of membranes. If we'll do, then first means in between the nucleus and perinuclear space, which one will be first, um, and which one will be in second position while coming from outside to inside.
If you see the typical structure of nucleus cell itself, from coming outside of a cell, you are coming outside, moving from outside to inside. So first
Yes,
of all, you
ma'am.
will encounter the cell membrane. Then you are moving
Yes,
inside the cytoplasm. Then you are encountering the nucleus. Nucleus has two membrane, outer membrane and inner membrane. Between both the membrane, the space which is present between two membranes, that is called perinuclear
yes,
space, right? So outer
yes.
membrane you will first encounter, then perinuclear space will come, then inner membrane will come.
Then
Okay?
inner. Acha,
Yeah.
okay, ma'am. Thank you.
Okay. Okay. I hope you have learned some new things today, and keep on reading the topic. And it's okay. We can take some time, but it is essential that you need to understand the things. You need to cooperate with yourself. If you not study, that
मैम
is your loss itself. Okay. So study sincerely. Yeah.
मैम आई हैव अ डाउट
Yeah. Yeah. Please. Please.
मैम सेल मेम्ब्रेन इज सेमी परमेबल और selectively permeable.
Yes, this was mine too.
Cell membrane, some places it is written like semi-permeable, some places it is written like selectively permeable. Semi-permeable, the term itself, it tells that means it is having some porous space, and between that pore, some molecules can pass freely, some molecules cannot pass because of the pore size. Okay. उसके पोर छोटे बड़े होने के कारण जो छोटे मॉलिक्यूल्स होते हैं वो इजीली पास हो जाते हैं और जो बड़े मॉलिक्यूल्स होते हैं वो नहीं हो पाते, राइट? सो बिकॉज ऑफ दैट रीजन इट इज कॉल्ड एज semi permeable. Okay, that only small molecule can pass through it, not the bigger one. Selectively permeable means it has the permeability to certain kind of molecule based on the chemical nature. Okay, based on the chemical nature, that which kind of molecule that is, which means whether it is a lipid, whether it is a protein, whether it is a carbohydrate, based on that cell will either allow the molecule to pass on freely or not. Okay, so you can tell the cell membrane as selectively permeable also, semi permeable, so no issue. Okay, because cell membrane is consisting of, everywhere cell membrane is consisting of lipids. Means the lipid bilayer is in the center, and towards both the sides your hydrophilic thing is there. Means outside of the cell facing surface is having hydrophilic, the cell facing cell that cytoplasm facing surface is also hydrophilic, but the in between layer is hydrophobic. Because of that region, whichever molecule is hydrophilic, so it will only let enter, means only let the hydrophobic molecule, that is a lipid kind of thing, to pass through it easily. and water can easily pass because water is very small molecules. Okay, and some ions which are smaller in nature and we don't have that much complexity in chemical properties, complex chemical properties, they can also pass freely. Okay, apart from that, there are specific pumps, there are specific channels, there are specific carrier molecules present over the membrane which will let the molecules inside and outside. Okay, is it clear? Is it clear?
Yes, ma'am.
Okay, do you have any other questions? We need to sign off now. It's already late. We have another class scheduled. Okay. So I'm ending the session today here.
Ma'am,
Thank you
next
so
class,
much.
ma'am, 1 minute, ma'am. Next class, what will be the topic? Hello.
Since today topic is not
Oh,
yeah, regarding the next class, it seems we will continuing this particular topic because the cell biology is not yet over. So you will be having 4 classes. I forgot to mention you. You will be having 4 classes per week for plant sciences. Okay, schedule will be shared with you very soon in one day or 2 day, and and the timing of the class will be varying. Okay, and don't worry, it will be according to your convenience also. And but mostly it will be in evening, mostly. But in sometimes if we also having some means, so if some mentor is busy because of some reason, we may alter the class timing, but that will previously will inform you, no worry for that. And which day which topic we are going to cover, we'll be sending you the complete schedule in 1 or 2 days. Okay, so based on that schedule, you need to prepare it beforehand, you need to study the topic beforehand, and like that. Okay, so next class, I'll be telling you when the next class will be, don't worry. Okay,
okay, ma'am.
anything
Thank you.
else?
No.
Okay, thank you for patient hearing. Read the topic once again and try some of the questions. After reading the chapter only, try the questions, that is good, because you have freshly read the topic, so you can try some questions. Okay, 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 then bye bye, good night.
Good night, ma'am.